All right, I think we can go ahead and get started. So, hello everyone, welcome to today's webinar. Just a brief introduction of myself. My name is Yuan, and I am a content marketing scientist here at Biocytogen Boston Corporation, and I will be moderating today's session. And I'm also joined by two panelists for today's webinar from the Biocytogen team, Dr. Yi Yang and Dr. Natalie Marchi. So Dr. Yang and Dr. Marchi, would you mind saying a quick hello to everyone and telling everyone a little bit about yourselves? Um, I think we can start with Dr. Yang. Yeah.
Hello, everybody. This is Yang. You can call me Benny. I'm the CSO of Biocytogen, and I joined the company in 2016, and I'm leading the drug discovery at Biocytogen. Very nice to meet you.
Thank you, Dr. Yang and Dr. Marchi.
Yes, hello everyone. I've been with Biocytogen for almost 3 years. I just transitioned from the CRO services to the licensing team as a business development manager. So I look forward to hopefully meeting you and speaking with you in the future.
All right, thank you both. We're glad to have you with us today. So, okay, now I would like to introduce our speaker for today's webinar, Dr. Wanbo Tang. So Dr. Tang graduated from the Academy of Military Medical Sciences in 2021, where his PhD work focused on the development of embryonic hematopoietic stem cells. And Dr. Tang has extensive industry experience in tumor immunology and drug development. And here at Biocytogen, he has been working on T-cell receptor or TCR-mimic antibody discovery, and he is mainly responsible for targets like KRAS, EFP, and survivin. So for today's webinar, Dr. Tang is going to walk us through Biocytogen's antibody discovery platforms with a focus on our innovative platform for generating fully human TCR-mimic antibodies. And he will explain how those TCR-mimic antibodies can overcome the limitations of traditional antibodies by targeting intracellular antigens. And he will share some case studies showing how these antibodies can facilitate the development of safer and more effective cancer immunotherapies. So before we jump into the presentation, just a quick heads up, the Q&A box is open, so feel free to drop your questions there during the presentation, and we will do our best to get to as many questions as we can during the Q&A session, which is after the presentation. All right, so without further delay, I will hand it over to Dr. Tang.
Hello everyone, I'm Dr. Tang, and I'm in charge of TCR-mimic projects in Biocytogen. And today I will introduce our antibody platform and our TCR-mimic platform. First, let me give a brief introduction of Biocytogen antibody discovery platforms. Based on our RenMice, we have developed 7 platforms to establish for the human antibody library, like monoclonal antibody platform, which are based on RenMice. Bispecific and bispecific ADC platform are based on RenMice. Nanobody platform is based on the RenMice. And also, we have TCR-mimic, and GPCR platform. Today, we will focus on the TCR-mimic antibody platform. Then, what are the differences and characteristics between RenMap, RenLight, and RenNano? I will describe them below. RenMice have complete human antibody heavy and light chain variable regions in situ replacement, and the mouse constant region is reserved to maintain mice antibody genes regulatory functions maximally. In situ replacement of variable regions have the advantage of higher accuracy and fewer uncertainties. When we get sequences after immunizing the mice, we can replace the mouse constant region by human constant region. And then we will have the fully human monoclonal antibodies with high diversity, high affinity, and low immunogenicity. On this basis, we developed a common light chain RenMice to avoid mismatch and easy to assemble into bispecific antibodies. Mismatch and purity are still sticky issues in the field of bispecific antibody expression, and RenLight is a perfect solution to solve these problems. Also, we engineered fully human nanobody mice with a single VHH domain to recognize antigen. It is heavy chain only antibody. Nanobody has smaller size, meaning it has better tissue penetration, like tumor and brain. Also, nanobody is flexible for multifunctional molecule assembly. Now, our RenMice are well recognized by worldwide partners. One of the major challenges during antibody development is immunogenicity. If antigenic epitopes are too homologous between human and mouse, the antigen immunogenicity will be low and it will be difficult to obtain antibodies. More than two-thirds of 150 targets estimated by us had more than 50% homology, and some is over 85%. For targets with high human and mouse homology, Biocytogen's target kill RenMice can greatly increase the probability of obtaining antibodies. This is a summary of our internal data. This is wild type RenMice, and this is target kill RenMice. We can get higher numbers of antibody sequences from target kill RenMice. Besides, we can provide a whole industry chain service from antibody discovery to pharmacology validation to collaborative clinical trials. Also, we have flexible business model and welcome for partners. Here shows a list of our developed projects. Now we have more than 900 target projects with 400,000 of fully human sequences in multiple therapeutic fields, including oncology, inflammation and autoimmunity, metabolism, and so on. Besides antibody discovery, our platform offers antibody engineering like antibody isoform switching and FC engineering, developability assessment like in silico analysis, stress study, and affinity maturation, and the antibody format includes ADC conjugation and bispecific antibody assembly and expression. Also, our CMC department can help with large-scale protein expression and purification, stable cell line development, monoclonal antibody process development like cell surface, cell culture process, purification process, ADC formation, and analysis methods development. Pharmacology is also an experiments department of Biocytogen and well received by worldwide partners. We have various mouse and disease models, like immune deficient model and reconstruction model, PK/PD study for antibodies pharmacology capability verification, and non-GLP tox study. This is an introduction about our antibody products and platforms. And next, I will introduce the TCR-mimic platform. And I will introduce the platform overview and case study. Here, we represent the entire drug targets library by an iceberg. Until now, most of approved drugs are membrane-associated proteins, accounting for about 27% of protein, like the iceberg on the surface. And Biocytogen has developed RenMice his platform to target these antigens. Meanwhile, most of membrane-associated proteins are tumor-associated antigens. It means antigens expressed in tumor cell as well as in normal tissue. While developing tumor-associated targets, it is necessary to screen for targets that are differentially expressed in normal tissues and tumors, which result in fewer targets suitable for development. However, 73% of protein are intracellular proteins, like the iceberg below the surface, and conventional drugs cannot target them. And they are all TCR targets. These targets include tissue differentiation antigens, cancer germline antigens, endogenous retrovirus, viral antigens, and new antigens. These antigens are not or poorly expressed in normal tissues and are ideal targets for drug development. It is reported that 12 of top 20 ideal cancer antigens are intracellular proteins. It means intracellular proteins have huge unmet clinical need and considerable development value. So how to develop intracellular targets? In 2022, FDA approved the first TCR therapy for uveal melanoma targeting GP100 and HLA-2 complexes from Immunocore. It seems TCR therapy is a viable approach. The epitope of intracellular proteins targeted by TCR therapy is peptide HLA complexes. Human leukocyte antigen, also known as MHC, is expressed on the surface of different cells and plays a key role in antigen presentation and immune signaling. MHC1 is a cell membrane glycoprotein consisting of alpha chain containing 3 immunoglobulin structural domains, the alpha-1, alpha-2, and alpha-3, and beta-2 microglobulin, the beta-2m. MHC2 is a membrane glycoprotein consisting of an alpha and a beta chain. MHC1 molecules are expressed by almost all cell types and allow for sampling of the endogenous cellular protein to be displayed for surveillance by CTAs, which then eliminate infected or transformed cells. The MHC2 pathway is found in antigen-presenting cells, such as dendritic cells, macrophages, B cells, that display exogenous peptides to help CD4 positive T lymphocytes, which support the development of cytotoxic and humoral immune responses. MHC1 molecules have closed-ended peptide binding groove and can typically bind diverse peptides, 8 to 12 amino acids in length for presentation to CD8 positive CTLs. By contrast, MHC2 molecules do not have closed-ended grooves and can bind longer peptides up to 13 to 28 amino acids in length for presentation to CD4 positive helper T lymphocytes. MHC1 and MHC2 peptide complexes are recognized by specific TCR receptors, also known TCR, with the aid of CD8 and CD4 co-receptors respectively. Here shows the process and the presentation of peptide HLA complex. Let's take an example of the surface protein mesothelin and the intracellular protein NYESO1. For mesothelin, ribosomes translate transcribed RNA into proteins in the cytoplasm, which are then shuttled to the endoplasmic reticulum for proper folding. Newly folded proteins then move into Golgi complex, where they are exported to the cell surface. Well, whether the surface protein or intracellular protein, they can be degraded into linear peptide, where peptide fragments by proteasome and loaded onto a HLA molecule. Then loaded peptide HLA complexes are delivered to the cell surface where Golgi complex recognized by TCR. Besides TCR, TCR engager like emetic, TCR-mimic antibody, and STAR-T or TCRMT can target peptide HLA complex. Compared to natural TCR, TCR-mimic antibody has advantages of higher affinity and easier to screen. The affinity of TCR-mimic antibody is nanomole to pmol. Both TCR and TCR-mimic can be assembled for cell therapy and TCR engager. So TCR-mimic antibody is a very promising treatment option. One of the difficulties in TCR-mimic antibody discovery is that the epitope is peptide HLA complex. If we use regular humanized mice, it will produce many antibodies targeting HLA rather than the peptide HLA complex. So, to get the sequences that more precisely target peptide HLA complex, we introduce HLA into RenMice, greatly increase the probability of obtaining antibodies targeting peptide HLA complex. This is the process of Biocytogen fully human TCR-mimic antibody discovery. Through immunizing HLA mice and high-throughput screening, we can quickly obtain fully human TCR-mimic antibodies with low immunogenicity and matured affinity in vivo. Also, these antibodies are naturally paired and maintain high diversity. Here are the immunization serum titer results of two projects, KRAS G12V HLA11 and MAGE-A4 HLA-A2. The results show that immunization serum generate dose-dependent immune response to its antigen peptide HLA complex rather than the control peptide HLA complex. Here shows heavy and light chain germline usage analysis and NSGAN's results of 3 TCR-mimic projects in Biocytogen. The inner circle is the heavy chain germline usage analysis, and the outer circle is light chain analysis. The same color means that the light and heavy chains are paired with each other, and the size of sector represents the number of clones. We can know that all 3 projects have highly genetic diversity. NSGAN is a site-directed mutagenesis technique that sequentially replaces the amino acids on a protein with alanine. The advantage is that the reactive group on the side chain is removed and replaced with a small, no other functional methyl group, which has less effect on the protein structure. It is a method that alters the surface residues without altering the three-dimensional structure of protein. The alanine scan experiment enables preliminary inferences about the antibody binding to each amino acid on the epitope, and the results show that Biocytogen TCR-mimic antibodies have diverse binding motif. And the sequence is the first step. Another problem for intracellular target is how to solve the low antigen abundance. Many papers and posters list the abundance of intracellular targets, and most of them are in the range of 10 to 500, and especially the pHLA mutation has only 2.4 copy per cell. As we know, CAR-T needs high antigen density, so T-cell engager and TCR-T therapy may be better for intracellular targets. Now, we have 4 types of targets: tumor-associated antigens like WT1, GB100, and AFP, and cancer-testis antigen like NY1 and MAGE4. Mutation targets like KRAS and p53, and virus target the LAMP2. We also offer customized antibody discovery with any potential target. The next is the case study, and the first is the GP100 project. As I introduced above, Kimmtrak, also known as terbinafusp, is the first approved treatment option for HLA-2 metastatic uveal melanoma, with a proven benefit in overall survival. It is a TCR bispecific fusion protein that contains a soluble TCR targeting GB100 and HLA-2 and a CD3 single-chain variable fragment to redirect T cells to lysis tumors expressing GB100 and HLA-2 complex. This is a side view of human eyeball. The wall of human eyeball consists of 3 layers, and the middle layer is the uveal, which is rich in blood and pigment. Uveal melanoma is the most common intraocular malignancy in adults. Up to 50% of patients with uveal melanoma develop metastasis, and the prognosis for these patients is extremely poor. Clinical benefit of anti-PD-1, PD-L1, or anti-CTLA-4 antibodies has been disappointing, maybe due to a low PD-L1 expression and low mutational burden. It is reported that more than 82% of metastatic uveal melanoma cells specifically overexpress the GB100 and do not overexpress it in normal tissues. And we found GB100 expressed only in melanocytes in normal tissues, and is also highly expressed in melanoma. So GP100 is a highly specific intracellular target. After immunization and beacon discovery, we can get some clones with high affinity and high specificity, targeting GP100 and HLA-2 complex. And we assemble one clone with CD3 to get a T-cell engager, and we perform multi-concentration binding assay. Positive control is the Kimmtrak analog, and we use the same CD3 as Kimmtrak. And the results show that the binding EC50 of our TCR-mimic CD3 T-cell engager is superior to the terbinafusp analog. To test the ability of activating T cells, we co-culture the antibody with tumor cell and jerked NF80 luciferase cells. We use the A2058 as the negative control tumor cell because it is GB100 positive but HLA-2 negative, and we use HLA-2 knock-in A2058 as positive tumor cell. And the results show that both TCR-mimic TC3 T-cell engager and terbinafusp analog can active T cells when co-culturing with HLA-2 knock-in cells, but do not respond to the negative control. And the EC50 shows that TCR-mimic TCR engager showed enhanced signaling than turbinate analog. We next examine the function of TCR engager in primary T cells. Negative control group is target cell HLA-A2 knock-in A2058 cell only, and the T cell plus target cell without antibody. And the tumor grew normally in these two negative control groups. Our TCR-mimic CD3 TCR engager and turbinate analog both show excellent tumor suppression at 0.1 nanomolar. But our TCR-mimic CD3 TCR engager exhibits better effect function than turbinate analog in vitro. In addition to GB100 project, we also screened TCR-mimic antibodies against the KRAS G12V and A11 complex and assemble it with CD3 to obtain KRAS TCR engager. And we use InQSight to test the TCR engager cytotoxicity in vitro. The green cell is KRAS G12V positive and A11 positive mutant cells. Use the positive control here. And the red cell are G12V negative but A11 positive, used as negative control here. And the gray cell is the T cell. And we can see that the T cells are redirected by KRAS T cell engager to kill the green positive tumor cells, but not the red negative cells, indicating specificity of our KRAS TCR-mimic antibody. And another application for TCR-mimic antibody is cell therapy, and I will take NYES1 as an example. NYES1 is the most popular TCR-T therapy target. It is highly expressed in many tumors and only expressed in testes of normal people, which makes it an ideal target. We perform antibody discovery targeting this peptide and HLA-2. After immunization and beacon selection, we obtain many sequences. They specifically bind with NYS1 positive cells, but not with the no peptide or irrelevant peptide positive cells. And our TCR-mimic antibody affinity is nanomolar level, and the red point is positive control. It is an affinity-enhanced TCR-mimic antibody. Regeneron have developed TCR for NYS1. Here shows the potential of target peptides predicted by Regeneron for NYS1. They engineered TCR to enhance affinity in vitro, but this leads to binding with off-target peptide URB1 and this peptide. In contrast, our antibodies hardly bind off-target peptide, as our TCR-mimic antibodies affinity mature in vivo and no need for in vitro affinity modification. We also perform anion scan and found each of our antibody has multiple binding sites. We all know that CAR and TCR have different components, which leads to their different sensitivity to antigens. And this article assembled TCRm by replacing TCR variable region with CAR variable region and test the response of TCRm with CAR-T on cells with different antigen abundance. And the different degrees of migration of flow plots represent different EGFR antigen abundance in A431 cells. and even gamma release and carrying experiments were performed by these cells. In the CAR-T group and these two groups, we can see that gamma release was lower in low antigen abundance group than that in high antigen abundance. In contrast, in TCR-mimic in TCR-MT group, there was little difference in gamma release between high and low antigen abundance group. A similar situation was observed in the carrying experiments, where CAR-T was weak in carrying cells with low antigen abundance, but TCR-MT was equally strong in carrying cells with high and low antigen abundance. It means TCR-MT may exhibit higher sensitivity to antigen-low tumors than CAR-T. To evaluate the carrying ability of CAR and TCR at intracellular targets, we engineered the same TCR-mimic sequence into CAR-T and TCR-MT, respectively. We use HLA-2 cells pulsed with different concentration of peptides to mimic target cells with different antigen abundance. When loaded with low concentrations of peptides, which means low antigen abundance, and the TCR-MT show more potent carrying activity than CAR-T, whether in 4-hour or 20-hour, and whether the ET ratio 5:1 or 1:1. TCR-MT showed stronger carrying activity. Does the TCR-MT also show antigen-specific response to natural target cells? We use 3 HLA-2 positive natural cell lines, U266B1 and SIH1755 express NY-ESO1 and HLA-2, but HCT116 do not express NY-ESO1. We could culture TCR-MT with these 3 tumor cells separately, and we collect T cells and tumor cells to detect activation and cytotoxicity, respectively. The percentage of CD69 in CD3 positive T cells show that TCR-MT can only be activated by positive cell U266B1 and NCH1755 and show no response to HCT116. Similarly, TCR-MT show dose-dependent cytotoxic activity to positive cells, but do not elicit the negative control HCT116. Furthermore, we engineered the TCR-mimic sequence into circuit NFAT luciferase to assemble TCR-mimic circuit reporter cell line, and we could culture the TCR-mimic circuit reporter cells with these 3 natural cell lines, and results show that TCR-mimic circuit reporter cells can be activated by 2 positive cells, but do not respond to the negative control HCT116. And since these 3 cell lines may not express the potential of target peptides mentioned above, and we use HLA-2 cells loaded with potential of target peptides for co-culture experiments, and the results show that our TCR-mimic circuit reporter cell can only be activated by NY-ESO1 pulse cells instead of potential of target peptide URB1 and MAGEH1 and so on, indicating our sequence high specificity and safety. Then we use myeloma cell U266B1 to test in vivo. We inject the U266B1 cell subcutaneously in TBNK deficient BNDG mice, and when the tumor grew to 100 to 150, we performed a single administration of TCRmT and CAR-T. Here is the results, and the red line is TCRmT group, and the green one is CAR-T group. Compared with PBS and MOC-T, CAR-T cells cannot suppress the tumor growth, while our TCRmT quickly reduce tumor volume in 2 weeks. After clearance of U266B1, we use another HLA2 positive and NYES01 positive cell, A375, to perform a challenge assay. We implanted A375 in another side of TCRmT treated and no treated mouse, and the results show that the tumor in TCRmT group cannot grow. It means our TCRmT can survive in vivo for a long time and continue to suppress tumor growth. At the same time, we have some projects completed for external licensing. Our WT1 project has licensed out to Biopharma for co-development. They assemble WT1 with CD3 and 4-1BB to get a tri-specific T-cell engager, and their tri-specific antibodies results are shown here. And the WT1 CD3 benchmark is RG6007, developed by Roche. And tri-specific antibodies also show the stronger on-target T-cell proliferation than benchmark, and dose-dependent tumor suppression effects. We also reached agreement on TCR-mimic antibodies evaluation and license with Biocopy, Fan Immune, and so on. And the Biocytogen TCR-mimic platform is well recognized by partners. Above all, Biocytogen's TCR-mimic antibody platform has its advantages. First, depending on RenMice and the Beacon platform, we can do faster and high throughput antibody screening of known and customized target. Secondly, TCR-mimic antibody is more convenient to screen than TCR, and the antibody has great specificity and high affinity, and do not need extra sequence modification, avoiding potential off-target risk. Thirdly, TCR-mimic antibodies have more application like T-cell engager and cell therapy, and we have RenMice with common light chain. It has an advantage for assembling therapeutic T cells with bifunctional or dual epitopes. And that's all. Thank you. Welcome for any questions and advice.
Thank you, Dr. Tang, for the insightful presentation. I think we've already got a few questions in the Q&A box, so let's take a look there. And to our audience, if you haven't sent in your questions yet, feel free to add them right now. So the first question here is, was the engineering of antigen at phenotype level or genotype level?
The antigen is genotype is we have several kinds of antigen, and we have did many projects, so we have tested many materials like cells, like protein, and like the mRNA. So we test the protein in vitro, and we think it's it is suitable to use.
And we've got another question here. Whether HLA RenMice mice are introduced with one HLA subtype or all HLA subtypes.
The HLA mice were introduced with one HLA subtype, and we have several HLA remap subtypes, not the all HLA subtype.
What are the materials used for immunization?
As I mentioned above, we have performed many projects, and we found that not a material antigen is suitable for our projects. So we have performed the cells, mRNA, and the protein for immunization. And we can get sequence for some we can easily to get sequence for some projects.
Thank you, Dr. Tang. And next question, does this mouse model have any phenotypes?
Yes, we have validated the mouse model phenotypes, and the phenotypes can be seen in the Biocytogen website. Yes, I guess every mouse model can be seen.
Next question here. Is the peptide HLA complex targeted by the JP100 project clone the same as KIMTRAC?
Yes, we targeted the same peptide as KIMTRAC.
Next question here. What is the aggregation risk potential of the TCR-mimic antibodies developed through your mice? Have you done stability studies or developability studies on these formats?
Actually, at Biocytogen, we do the stability test and the developability test for all the antibody clones. So in general, the TCR-mimic antibody do not have any potential issues because these are the conventional antibody recognize desired antigen, especially except that these antigen is not general like it's extracellular proteins, but these are the HLA peptide complex. But because these antibodies are derived from the mice, so they go through all the in vivo maturation and also any sequence has liability. This will be screened out during the development of the immune response. So this ensures the in vivo derived binders has good properties in terms of physical chemical properties and the developability. But in addition, when we have the good sequence, we will do all the in-house examination to see whether there's any potential issues we need to further optimize. So in general, I think the TCR-mimic antibody, they are as you can treat this antibody as a conventional antibody. They don't have any anything will cause our attention.
All right, thanks, Dr. Yang, for the detailed answer. And here the next question, does Biocytogen make their own pHLA antigen complexes or if you use an external company?
We can make the peptide HLA antigen ourselves, and we also can purchase it external company, and it depends on what you need.
Okay, I think we covered all the questions here. So thanks everyone for sending those questions in, and a big thank you to Dr. Tang for the great presentation, and to our panelists, Dr. Yang and Dr. Marchi for sharing their insights. Oh, we have another question actually. So what's the half-life of your BITE?
We did not test the half-life, but we tested the winding. Stability of our protein, and the data is very good, and I think the half-life of our BITE is as long as the KIMTRAC, and the KIMTRAC half-life time is 20 hour is about 20 hour, and I think our bite is equal to that.
All right, awesome. Thank you, Dr. Tang. I think that's all the questions. Again, just a quick reminder, if you missed anything, this webinar will be available to watch on demand. You can request it on Biocytogen's website, or you can find it on our YouTube channel. And if you have any additional questions, feel free to reach out through our website, and we will be happy to follow up with you. And also feel free to check out our website and the LinkedIn page to learn more about our projects and assets. You can also search for, Ren Biologics or our RenMice platforms there to explore more targets of interest and our latest progress in antibody discovery. All right, I think that's it for today. Thanks again for joining us. Have a great rest of your day, and we hope to see you again soon. Thank you.
Please.
Thank you. Bye-bye.